Buon pomeriggio, sono Leonardo e faccio parte del gruppo di giovani volontari di BergamoScienza. Prima di iniziare vi chiedo un momento di attenzione e vi ricordo che collegandosi al sito bergamoscienza.it e cliccando sul pulsante live è possibile postare su Facebook e Twitter la diretta dell'evento, inviare domande ai relatori oppure invitare i propri amici e follower a seguire la conferenza in streaming. Le domande dal web vengono ricevute da noi volontari che ne proporremo una selezione durante il dibattito in sala.
Volunteers during the question and answer session. For simultaneous translation, you can scan the QR code that you find on the hall doors or projected behind me. The QR code will address you to the Converso web app where you can select the language from the scrolling menu. To listen to the translation, you can use your own earphones or ask for earphones to the volunteers in the room. I would like to welcome you to today's meeting, a new map of the ocean life, where we're going to listen from Chris Bowler from the Stazione Zoologica from Naples and Telmo Pievani from the Association of BergamoScienza introducing the conference. You are kindly asked to mute your mobile devices and take part in our initiatives. Thank you very much for listening and enjoy the conference. Thank you and good afternoon. Welcome on my side to you all. It's a great pleasure for me to introduce today's event because in a few moments we'll be diving into world oceans. This is what will happen in a short while, and we'll do this with a scientist who is dear to me, Chris Bowler, who has plenty of roles and positions. I cannot mention all of them. For the past 12 months, he has become the president of the Zoological Station of Naples. This station was founded in 1872 by Anton Don, Anton Don, dear friend of Darwin, who founded this station. Thanks to Darwin's help to carry out marine life research, and this was supposed also to confirm Darwinian theories in the oceans. For the past 12 months, Chris has been appointed the president of the Zoological Station. He isn't carrying out research there, he is carrying out research in Paris. He also worked for this station from 1994 to 24, and he also works in Paris. He's the director of CNRS. A lot of awards were won. He was awarded 2 ERC Advanced, very rare things in just 1 year, 2 awards. And another thing I'd like to mention is this very interesting project Chris is director of, Tara Oceans. You might check it online. Tara Oceans, a beautiful site with all the information on this project. It is a scientific schooner to observe the oceans. It has toured all the world oceans to take samples, to carry out observations, to study the invisible ocean biodiversity, diatoms, microorganisms, and surfing the site, I found a very interesting statement about Tara Oceans. In search of our ignorance. And I like it very much because the message is through this project, they could discover incredible things, a lot of things we did not expect, new species, unknown genes, and this is very good news. And then we have a least positive piece of news. A lot of microplastics is being found, and another bad piece of news is how sensitive this biodiversity is to changing temperature, to climate change, and the dangers we are running because of this. But the title is that we realize how ignorant we are about the biodiversity in oceans. We look from the upside and we see it as an even surface, but beyond the superficial level, it is plenty of life. Thank you very much. Chris now will have a speech, and then we have a question and answer session. Welcome.
Grazie Telmo, è un piacere di essere qui.
Thank you, Telmo. It is a great pleasure to be here.
Per fare confusione ai traduttori farò un po' in inglese e un po' in
I'll be speaking both Italian and English to confuse translators, but I'll be making my presentation in English, so it'll be quicker. And it is not a difficult presentation. Then I'll be answering the questions in Italian so that I can have a conversation with you, which can be made easier for everybody. Now I switch to English.
Well, the title that you gave me is Una nuova mappa della vita nell'oceano. So I will try and talk about this sort of context of my presentation. I will talk in particular about Tara Oceans and this boat that you see here behind, this sailboat on which we do our research today with the foundation Tara Ocean. So, exploration of the oceans through the centuries, there's nothing new, of course. We have been exploring for many centuries new territories. Most of the time, the aim of this exploration was to find new resources, to discover new lands, new resources, new kinds of plants, new kinds of animals that we can exploit in our economy as resources in agriculture and so on. There were also some crazy adventurers who wanted to discover the North Pole, discover the South Pole, to go to the extremities of the world. But these days we know pretty well the surface of our planet at least, and we have many different tools that we can use to explore, to study the ocean. We have very large oceanographic ships full of all kinds of special equipment. We have drones, which we can put in the atmosphere, drones we can put in the water. We have satellites to observe the oceans from space. We have all kinds of ways to study. So the way that we do exploration has really changed over the centuries. And what I'm going to talk to you today, I'm going to talk about life in the ocean. Okay? So no physics, no chemistry, only biology. At least mainly. So when we think about life in the ocean, you may think, well, where is the life in the ocean? Okay, when we go for a walk in a forest, it's easy to see the life. We have trees, we have plants, we have animals, we have insects all around us as we walk on land. But in the ocean, it can often seem like there is no life. Okay. The magic ingredient to look for life in the ocean is to take a microscope. Then when you take a microscope, a whole world opens up with all these amazing forms of life of all different shapes and sizes that do all kinds of important things, as you will see in my presentation. So these are microscopic. They have many different sizes. You can fit many of them onto the head of a pin. They have all kinds of different shapes, and they have all kinds of evolutionary histories. So here we have a tree of life, where we see all different kinds of life forms. And we animals, we are down at the bottom here, if you can see my pointer. We animals are down in this, sitting in this branch here. And our closest neighbor, our nearest neighbor, are sponges, le spugne. So this is the evolutionary distance between us and sponges, all right? But you see all of this incredible diversity of life, all of which is present in the ocean, in this microscopic world that we want to explore. So it's really true that in every drop of seawater, you have all the evolution of life that is present that you can explore and try to learn more about. So you have the origins of life, and you have all kinds of amazing life forms that are in this microscopic world. And they do all kinds of important things. First of all, in terms of biomass in the ocean, the amount of life in the ocean. If you just imagine taking a giant net and scooping up all of the organisms in the ocean, all of the whales and the dolphins and the fish and the microorganisms. If you do this and then you add up what you find in fact that this invisible life represents 2/3 of life in the ocean. Okay? The other 1/3 are the fish, the whales, the dolphins, the corals, things you see on TV. But clearly on television, there is 2/3 of life in the ocean which we don't usually see. And this is the plankton, the invisible multitude of life in the ocean, with all kinds of important activities. They are the basis of the food webs of the ocean. So if there is no plankton, there is no fish, there are no whales, there are no anything else, because it's the base of all life in the ocean. Many of these planktonic forms, they do photosynthesis, so they are microscopic plants. And just like plants on land, they take carbon dioxide out of the atmosphere, and they convert it into life, organic biomass, which feeds the food chain, and they also generate oxygen, which we breathe. In fact, every second breath that you breathe is oxygen coming from the photosynthesis in the ocean. Okay, so photosynthesis is not only forests and trees on land, but there is just as much in the ocean as well, thanks to these microscopic organisms. So this is really clearly very important to manage the state of our planet. In addition, because the ocean is very deep, when these organisms die, some of them will go to the bottom of the ocean. And this is a very sort of precipitation of kind of plankton, which over the centuries goes to the bottom of the ocean, and this in fact generates the oil and the gas that we burn today. So it's fossil fuel. The fossil is plankton for all of our oil and all of our gas. Okay? So this gives you an idea of how important this, we call it a carbon pump, which takes carbon dioxide from the atmosphere and takes it all the way to the bottom of the ocean and stores it for millions of years. This is our most powerful biological carbon pump on the planet, and it is our most powerful weapon against climate change, because it removes carbon dioxide out of the atmosphere, which is our biggest enemy when dealing with climate change. So this is a really important function that plankton do. In this way, they affect the climate, because climate is basically carbon dioxide in the atmosphere. But at the same time, plankton are also sensitive to climate change. So plankton change as the environment change, and this is something which we need to think about in detail and understand better. So in this first little summary, I want to end by saying that we can think of plankton as the ocean microbiome. Okay, you maybe heard of the human microbiome, that we have gut flora, microbes in our intestines that ensure the digestion of our food. We have microbes all over us, which ensure our state of health and also ensure our state of well-being, actually. Exactly in the ocean, these microbes, these microscopic organisms, they play all kinds of essential roles in terms of recycling minerals, recycling nutrients, providing carbon that feeds the food chains, and also doing all kinds of important activities to remove all of the damaging substances that we throw at the ocean today, be it microplastics, be it other pollutants, be it tourism industry, aquaculture industry, and so on. So the microbes, I think we should really consider as being at the center of life in the ocean and the major determinants of the state of health of the ocean. So this is why we wanted to study plankton, okay? In the last few minutes, I've explained, hopefully, why microscopic life is important, and why we wanted to study them in the Tara Oceans project, which was began with a 4-year expedition around the world, as you see on the map. So this was the scientific activity of Tara Oceans. This was performed on a research sailboat called Tara, which you can see there. It's not a big research oceanographic vessel. It's only 36 meters. We have 14 people on board, 6 scientists, 6 sailors, 1 artist, and 1 journalist. So we cannot do too much, but this is what we do. I will explain in terms of our science. The Tara sailboat is owned by the Tara Ocean Foundation, which is owned by Agnès B., who is a fashion designer, a very famous fashion designer in Paris, in France, and she graciously gives some of her own money to finance the science that we perform. Okay? So in addition to the science that we perform, the foundation also spends a lot of time doing popularization of science. That's also why I'm here at your beautiful festival here in Bergamo. Education, contributing to the national curricula, and also influencing policymakers to try to improve the governance, the management of our oceans. So during this 4 years of expedition around the world, we wanted to study these microscopic plankton organisms. Let's just look for a second who is present in these communities to give you a better appreciation of what is present. One group of organisms we have, we can call them protists or single-celled eukaryotes. Quite complex cells, but they have all kinds of amazing morphologies that you can see here. Always in a single cell. Sometimes they form chains, sometimes not. They do all kinds of important things, including photosynthesis, but some of them are parasites, some of them are pathogens, all kinds of things. So in general, in an average liter of seawater, we can have up to 10 million organisms, okay, present in 1 liter of seawater, of these kinds of organisms. In addition to the protists, we have zooplankton. These are kind of the grazers of the ocean. These will eat like cows, like sheep. They eat the microscopic plants that are in the protist world, for example. Here, these are multicellular organisms. They're more complex organisms. Some of them look like little shrimps like this, just less than a millimeter in size. And these can be present at up to 100 organisms in an average liter of seawater. Okay? Then, getting smaller, we have lots and lots of bacteria. Some of these do photosynthesis, some do not do photosynthesis. They are recycling minerals, nutrients. These can be present at up to 1 billion cells per liter of seawater. And then, even smaller, we have the viruses. I don't need to explain how important, how powerful viruses are these days, even though they are incredibly tiny. They're incredibly important, and these can be present at up to 10 billion viral particles in a liter of seawater. Okay? So no panic, these are not dangerous for us. These are infecting other kinds of organisms in the plankton world. Some of them infect fish, some infect corals, but they are there really to infect the organisms within this microscopic community. So we have all of these different organisms present in this microscopic world, and we wanted to sample all of them. And we wanted to do this at a global level. Here maybe it's best if you watch this video to sort of see the science that we performed on board the vessel. These are plankton nets which would sample and concentrate particular kinds of plankton. You see it moves quite a bit. Even in Bergamo c'è mal di mare. Here's some zooplankton that we collected. This is a machine called a rosette, which we can send down 1,000 meters into the water, bringing back water samples, which we then filter, concentrate, and put onto membranes, which then we analyze afterwards. We have a small laboratory on board with microscopes and other kinds of special equipment where we can really see what the organisms actually look like as we are doing the sampling. We sample at night, we sample by day in order to capture all of the different kinds of life forms that are present within 1,000 meters of the water column at each place that we would sample. And typically we would stay for 2 days, 2.5 days in one place, sampling to collect all of these different kinds of life. Okay. So this was the wet part of the project, let's say, the beginning of the project from 2009 to 2013. We went around the world. We actually went 2 times around the world, first going through the temperate oceans, going first through the Mediterranean, the Red Sea, the Indian Ocean, the South Atlantic, into Antarctica, out towards Tahiti, Hawaii, back to San Diego, Panama Canal, New York, and back home in 2012. And then in 2013, we set out again to go around the Arctic Circle. All right, so Tara is in fact the first sailboat in history to go through the famous Northeast Passage and the Northwest Passage in the same year. Okay, not such a big record anymore because the ice is melting so much that it's actually quite easy to get to do that these days, sadly. So throughout this 4 years of sampling, we sampled at 210 sampling sites. We always did the same thing, which is very important to be able to compare one sample with everything else. So for example, to compare what we find in Arctic with what we find in the Maldives. This was really important for what we subsequently did. And at the end of this exercise, we had something like 40,000 samples, which we had collected and sent back to the labs for analysis. So here the work begins after the expedition, if you like. And just like the work on the boat was very standardized, also the work in the lab was also very standardized. We always did the same thing with all of these samples. So on the one side, a big, powerful system that we used to analyze the samples was microscopy, just looking at seeing what the organisms are. Here you can see a range of beautiful protists, each one single cell, each one a few microns in size, a few tens of microns with beautiful structures. But because we had many thousands of samples, we couldn't just look in a microscope and take pictures. We needed high throughput, high resolution methods. So we set up automated imaging systems to get through all of these systems, all of these samples. In parallel, what we did was DNA sequencing. Again, high throughput sequencing, just sequencing DNA in different ways to learn what is in these samples. And our basic goal was to ask a very simple question. Who is there? What do they do? With whom are they doing it? And why are they doing what they're doing? Okay, so we're trying to focus in onto an invisible world and interpret what is happening using the methods of microscopy and genomics. So we started to do this already quite some years ago, and we were very happy that we could publish the first results from the project in a special issue of Science magazine in 2015. We were very happy that they gave us 5 papers. We published 5 papers in this special issue of Science describing the first results of the project. They gave us the front cover, and we had a lot of news all around the world, including front page of the New York Times, talking about the things that we had discovered. So this is kind of the first inventory of life in the ocean at a global scale, and it represents life at the beginning of the 21st century, the life in the ocean at global scale. So what can we do with this data? Right? I'm going to give you a few examples of things we can do, going from simple to a little bit more complicated. But it's still very easy, don't worry. So first of all, let's ask some very, very basic questions. The kind of question that a 5-year-old child might ask, or a kind of question that an alien coming from space might ask us, to know about with whom do we share our planet, all right? Kind of questions: which plankton group is the most abundant in the ocean? Or which plankton group is the most diverse in the ocean? Okay, so really simple questions. So maybe you can start thinking what the answers might be. And while you're thinking, let's just take these questions and ask them for terrestrial ecosystems, the world around us, the world we can see. And ask these questions for these terrestrial systems. So in terms of abundance of organisms, well, if you look outside, the world is green, right? Outside of our cities, the land is green. And that is because, that is because the plants are the most abundant organisms on land, all right? They far dominate in terms of biomass anything else that we have in terrestrial ecosystems. You see animals at the bottom right. We are in the animals. We are not very important in terms of biomass. It's really the plants that are dominating life on land in terms of biomass. In terms of diversity of organisms, the answer is different. This diagram on the right shows diversity in terms of size of the organism, okay? And what you see here is that the insect is biggest. So in fact, the insects are the most diverse organisms on land, okay? We have hundreds of thousands of different species of insects. So the question that I'm asking here is, what is the equivalent of the plants and what is the equivalent of the insects in the ocean? Okay? I don't know if anybody wants to make an answer, but you'll probably be surprised in both cases as we were. The most abundant organisms in the ocean turn out to be organisms known as radiolarians. How many people have heard of radiolarians? 1, 2, 3, 4, okay. These are the most abundant organisms in the ocean, okay, in terms of biomass. We have known about radiolarians since 150 years. These beautiful drawings of Ernst Haeckel, made in the 1870s, show the amazing beauty of these single-celled organisms, okay? Each one tiny, few microns in size, with these beautiful architectures. So we know about these organisms since a long time. We just didn't appreciate their abundance, because when we sample the ocean, these kinds of organisms, they tend to break up, they tend to explode. So we haven't really quantified them very well in the past. But in our oceans, we used a camera which we put into the water column, and it took images, it took photographs of every organism that it found. And by doing this in situ imaging, we could reveal the amazing biomass of these radiolarians. Okay, so these are the equivalents of the plants in the ocean, right? That's take-home message number one, which probably is a surprise to all of you. It was a surprise to us. Now, in terms of diversity, the answer is very different. The answer in terms of diversity are diplonemids. Okay, so who has heard of diplonemids? Nobody. We never heard of diplonemids when we got this answer, okay? So, and we don't know much about diplonemids. They turn out to be the most diverse group of organisms in the ocean. They are sitting in the tree of life, close to important human pathogens like trypanosomes, leishmanias. So we assume that they are sort of parasites. But we don't have them in the lab, so we don't know exactly what they do. So any young people want to start a career in science, you should study the insects of the ocean, which are the diplonemids, okay? So those are, you know, some surprising answers to some really, really simple questions. What we can do is we can increase the complexity a bit more, because we have a huge data set, so we have information about viruses, about bacteria, about protists, about zooplankton, from hundreds of different places all around the world, at different depths in the water column. So we can take all of that data, and we can do a giant compute to see which organisms do you always find together in your samples, or which organisms do you never find together, okay? So in this way, we can define the social network, la rete sociale, of the plankton. We can define the Facebook of the ocean, of the plankton world, by trying to see who interacts with who, okay, in this invisible world. And this is the result of that. It doesn't look very nice, but it's incredibly informative. Each dot, each point is an organism. It could be a bacteria, or it could be a zooplankton, it could be something. And then the lines between 2 points tell us if they are always together in the same sample, we have a green line. And if they're never together, we have a red line. Okay, so we begin to define plankton that are very social, plankton that are very popular. You know, I don't know who you would say in Italy, the soccer players for the Italian team are these popular kinds of plankton that everybody loves, okay? Then you have other kinds of plankton which have the red lines, which are kind of the antisocial plankton, the kinds of plankton that nobody likes. These are the Putins, the Trumps, okay, I'll not say more, of the plankton world. So in this way, we can begin to look at interactions between the plankton, all right? And this is where we start to look at the ecology of these organisms. And just at one very simple level, if we just take all of the positive associations and all of the negative associations, we pull them apart, you know, the green ones, the red ones, and we see how many there are, we have quite a surprising result that the number of positive associations is much higher than the number of negative associations, which what that means in ecological terms, perhaps, is that cooperation is more important than competition. Okay? These organisms cooperate with each other, they interact with each other, and this appears to be more important than competing with each other. Okay? This was another surprise to us because we think, you know, the Darwinian ideas of the fight for survival, competition between the organisms, perhaps that's not really the case, at least not in this plankton world. In the plankton world, it's more about cooperation, working together as a community to ensure that the system works, right? That seems to be more important than screwing your neighbor, which is a nice, I think, message from the plankton world also for our own societies. So that was a sort of high-level result that we got by looking at these, this social network of the ocean. What you can also do, of course, is go and look for your favorite organism and see, well, what does it interact with? So if your favorite organism is somewhere one of these blue dots, you take it and you see, well, who is it interacting with? And here we can also learn all kinds of new things. So I give you one example of that with this organism called Symbiodinium. It's not very nice to look at, but this is the symbiont that we find in the coral reefs. This is what gives the color to the coral reefs, okay? Through symbiosis with the corals, it gives us the beautiful coral reefs that we all enjoy. And Symbiodinium also makes all kinds of symbioses with other kinds of animals, shellfish, mollusks, worms, snails, okay, which is always responsible for the color of these organisms. So we know about Symbiodinium forming symbioses with these organisms. That's why it's called Symbiodinium, symbiosis. So the question we wanted to ask is, well, do we find Symbiodinium in symbiosis with plankton? Not with these bigger organisms, but with plankton. And the answer, we were very surprised by looking at the social network of the plankton and then looking in our microscopy data, is indeed that Symbiodinium lives in symbiosis. It doesn't float around in the ocean as a naked cell, moving around from coral reef to coral reef, but it's found a house. It's found a house, and that house is another organism. It's an organism that is ciliated. You can see here, this is just a few tens of microns of size. It's very small. It has cilia on the outside, you can see very clearly. And inside this organism, these red and blue balls are Symbiodinium cells. Okay, so it's found a house to live in, protects itself from predators, pathogens, who knows what. And in this way, can travel around the ocean and then jump out and find coral reefs when it wants to do so. So these are some of the examples of some of the amazing discoveries that we have made using this incredible data set. Since the first paper in 2015, we have published many other papers as a group of scientists, and sometimes at very high level, sometimes they give us the front covers of the journals and so on. And just to give one summary slide of what we described in this plankton world, we have about 35,000 different kinds of bacteria containing about 47 million genes in total. We have more than 200,000 different types of viruses in the ocean. Before Tower Oceans, we only knew about 39 of them. So this shows you the amazing discovery that we have been making. And for the protists, we've described about 130,000 different types of protists, more than 10 times higher than the number of formally described species of marine protists. And about 1/3 of them, we have absolutely no idea what they are. Okay, so again, young people need to come into this field and try to tell us what this 1/3 of life in the ocean actually represents. we've also catalogued loads of genes from these organisms too. And then we were very happy to see in another very highly prestigious journal, Nature, earlier this year in June 2023, they actually wrote a little editorial about Tara's inspiring 20 years at sea. So that's a real big recognition for a scientist to be given this sort of accolade from the scientific journal. So we're very, very happy that we have achieved so much with this project on this little sailboat. So those are some of the things that we have done. I will not go into more technical detail about what we have done, but in the time that is left, I want to tell you about what we can do with this data with respect to climate change. We all care about climate change, we're all concerned, we're all worried about climate change, so is there anything that we can use this data for to tell us something about the state of the ocean today and maybe how the ocean will change in the future? So before I do that, let me give a little bit of context to this final part of the talk. Here what we have is the history of our planet in a 12-hour clock, okay, beginning at midnight with the origin of the Earth. And what we can see here is that photosynthesis was the first life to evolve, and it evolved around 2 o'clock, okay, somewhere here, at 2 o'clock. And then there wasn't that much happening until around maybe 10 o'clock in the evening up here, okay? 10 o'clock is when the animals first appeared in the ocean, the Cambrian explosion, you maybe heard. These multicellular animals suddenly started to appear in the oceans. And then following that, at about 11 o'clock, life went on land. First the plants went on land, and then the animals went on land, not online. And the dinosaurs appeared, well, what, about 11:30 or something, and then disappeared at around 11:45, okay? We, human beings, we appeared in this representation 2 seconds ago, okay? So our history on this planet has been 2 seconds since the emergence of the Homo genus. In this time, we have caused all kinds of damage. Even if we look in the last 100 years, we see what we call the great acceleration of human impact on our planet. Really, since the Second World War, we can see these plots sort of beginning in the 1950s. We have this exponential rise, be it of world population, be it carbon dioxide in the atmosphere, be it loss of tropical forests, whatever. We see this huge footprint of human activities on our planet. So we have become a superpower. Really, in every sense of the word, we have become a superpower. And our impact is also seen in the ocean. Here we have actual data. It's not simulations, it's not models, it's real data. Showing how sea surface temperature has increased since 1900. You see it's very clear, the trend. This is the increase of sea level since 1900, the loss of sea ice in Arctic, and here we have, in the bottom right, the acidification of the ocean, because CO2 mixes with water and makes carbonate and bicarbonate, and that's acid. And again, we can see with actual data how the pH is changing in the ocean. With data now since the 1980s, the data is very clear. So this is already very clear and is already alarming enough. But I wanted to show you this. This is what is happening this year. This is why it is so hot in this room today. It's because the temperature of the global ocean is about 1.5 degrees higher than it has ever been since records began. This is not the Adriatic, it's not even the Mediterranean, but it's the global ocean, the whole planetary ocean. I think this is 31st of August here, 31st of August 2023. You see, it's completely off scale since month of April. It's not behaving like it normally does. This is the average through several decades. So the temperature of the water is really increasing dramatically this year, and this is particularly alarming. And so this CO2, greenhouse gas, is not particularly surprising, but the speed is surprising. And this brings me to a bit back to biodiversity, so going from climate to biodiversity. And so biodiversity has not been changing slowly, steadily, increasing over time, but biodiversity changes over time. And biodiversity has been interrupted at least 5 times in the past. There have been 5 major events. The last one made kaput the dinosaurs at the end Cretaceous. And then there's been 4 events since the dinosaurs disappeared as well. So when we look at what happened, why did 95% of life disappear at these mass extinction events? The data is actually pretty clear in that these events are caused by massive disruptions in the carbon cycle. Okay, so it's all about carbon. It's all about where the carbon is. The carbon can be carbon dioxide in the atmosphere, greenhouse gas, very dangerous. The carbon can be in life, like us, and the carbon can be in the rocks. Calcare, limestone, is carbon, okay? And it's when the equilibrium changes between how much carbon is in the rocks, is in the life, is in the atmosphere, that we have these major disruptions of life on the planet, okay? And that's why what is happening today is particularly worrying when we look at carbon dioxide levels in the atmosphere. Today, we are at 420 parts per million. And as you see from this plot, going back 800,000 years, we haven't seen these sorts of levels for almost 1 million years of life on our planet. So we have high levels that are being reached, and we also have a very rapid increase, which are both quite worrying trends. So the question now then is where are we going? Are we going towards catastrophe, or is it? Are we going to be okay? The answer, we don't know. All right, nobody can really say that, but we can try to use scientific data to try to understand. Maybe predict better how the ocean is going to change in the future. And I have 3 examples of how we have used Tara Oceans to address changing climates, which I don't really have time to talk about, all of them, but let me tell you about evidence for the effects of ocean acidification in this one simple example that I show you here, where we're basically trying to analyze whether there's been any impact of ocean acidification in the last couple of 100 years. This is difficult to do because we don't have samples from the ocean before the Industrial Revolution. Okay, so it's difficult to compare samples from today with samples from the past. Except in one example, we have samples from this first oceanographic expedition on a ship called HMS Challenger in the 1870s, where they went out to study life in the ocean. They collected samples, brought them back, and these samples are stored in the Museum of Natural History in London. So we can have access to those samples, and the particular organism that we were interested to look at are organisms called foraminifera. Again, they're single-celled, beautiful, amazing cells, even though they're just tiny single cells. And we're interested in foraminifera because these shells that they make are calcified. They're made of calcare, of limestone, which we know is sensitive to pH, right? When you have calcare in your washing machine, you add lemon, you add acid, it disappears. So acidification potentially can damage these organisms. So we were interested to see, well, do we see changes in the calcification of these organisms over time? So in this example here, we're taking samples from the HMS Challenger from 1870s, and we're comparing them with samples from Tara. And in the blue color here, at the end, we're measuring the thickness of the calcium carbonate cell wall. And you can see in this one example, we have data from thousands of organisms. We can see how the thickness of the shells is really getting smaller as a consequence, presumably, of increased acidification in the ocean. So that's one of the few examples we really have that really shows how ocean acidification is affecting organisms in the ocean. Okay, it's one of the very few examples that we have. And then another example, maybe in closing, is what we can learn about the future ocean. How will the ocean change in the future? Is there anything we can learn from the plankton that we have today to project how they may change in the future? And so to do this, we first generated, kind of global maps of the diversity of different kinds of plankton organisms at global scale. As you can see here, these are photosynthetic bacteria, for example, and we plot out the diversity in terms of color. So in each of these 6 examples, we can see diversity is highest in the tropics, and then it goes down towards Arctic and Antarctic, okay? This is latitudinal gradient of biodiversity, which we see on land, and we now see for the first time in the ocean with this actual data. So we can take these maps of plankton diversity distributions from today, at the beginning of the 21st century, and we can throw them into the ocean, which we predict to have in 100 years' time. This is ocean predicted by the IPCC models, okay? The International Panel of Climate Change has made these projections of how the ocean will change in the future. For example, in terms of temperature changes. This is in terms of photosynthesis on the right side. So we can take today's plankton, throw them into this future ocean in the computer, because it's only in the computer. It's not a real ocean. And then see how is the plankton going to behave in terms of diversity. And here we're plotting anomalies of diversity of each of these 6 different kinds of plankton at a global scale. And what we basically see is that the anomalies are likely to be strongest at the higher latitudes, going towards Arctic and Antarctic, which we can plot by latitude here, on the left going from Arctic, equator, Antarctic. These are the diversity anomalies of different groups. And we can see that where we predict the anomalies to be highest are regions where the export of carbon is the strongest, is the most important. So this is a really important defense against climate change because it takes carbon dioxide out of the atmosphere into the ocean. These are the areas where our fishing industry is particularly intense. Okay, so because the fish eat the plankton, our fishing industry is maybe likely to be affected by these anomalies in plankton diversity as well. And then on the final panel is a map showing marine protected areas in the global ocean. And again, we have many areas of marine protected areas at these latitudes where we predict changes in plankton diversity to occur. So exactly the sites where we try to preserve, to look after the diversity, the biodiversity of the oceans, are maybe the sites that are maybe most at risk from these changes. So that is another way that we can sort of use the data from today to try to predict how the ocean may change in the future. And this is obviously very important to try to mitigate the changes, the damage that can potentially occur in the future. I think I will maybe end the science with that and move to conclusions. Do I have, what is the time? Okay, maybe 1 minute. Okay. Going a little bit beyond the science. So I'm a scientist. What I do is science. That's my passion. That's what I know most about. But we also have to translate our science into policy. We have to translate it in a way that politicians can understand, in a way that change can be made, okay? And the Tara Ocean Foundation spends a lot of time trying to do that, on the one hand engaging with young people, and on the other engaging with very powerful people on the planet. So this is Ban Ki-moon, the Director General of United Nations, who came on board a few years ago. And we were particularly active during the COP21 climate change conference in Paris, where we came together with other organizations and formed an organization called the Ocean Climate Platform. And we all decided to go to the COP21 in Paris and talk about the ocean, which had never been done before. The COP21 was the first COP when anybody talked about the ocean. Collectively, you see many logos that are familiar, WWF, IUCN, many others that you will know, that we all came together with the Ocean Climate Platform, and we made a lot of noise about the ocean at the COP conference in Paris. And we were successful in the sense that the treaty, the Paris Treaty, for the first time mentions the ocean in the text. Took 21 years, COP21, 21 years to recognize the importance of the ocean for the climate debate. And it's only in the preamble of the Paris Treaty that mentions the word ocean and the importance of the biodiversity within it. So this is a nice example of how collectively coming together we can try to bring the best science onto the table and make change all the way up to the highest level. And these days the COP conferences are almost 50% ocean, 50% terrestrial. So it's really been a very great collective success that we managed to achieve. So I'll maybe stop there and we can move to the questions. Just to thank many organizations involved in this work. We're maybe 25 laboratories worldwide, including Stazione Zoologica in Naples. Milano Bicocca was also involved in the project. Our private sponsors, from Agnès B to the Prince Albert II of Monaco Foundation, have helped us. And our funders of the research include European funding and so on. So thank you to all of them for their support of this project, and thank you to you for your attention.
Grazie. Thank you very much. Thank you for this talk, for your research and the commitment. Now we have some time for the questions. I'm going to ask you to please show up so that the mics can come. And I'd like to ask you something straight away. We can pass to Italian.
Hai ripetuto spesso, a parte che è fantastico scoprire che noi mammiferi
It's amazing to see that in terms of diversity, us mammals are nothing compared to diplonemids that we saw before. And you often repeated that you ask yourselves simple questions, how much biodiversity there is, why, and so on. So basically what you do is basic research, based on curiosity to understand what's out there. Could you explain why this is so important, why basic research is so important? You showed how important it is to understand climate change. But we know that from this basic research you can have amazing discoveries such as gene editing in bacteria. Why is it important to finance this? Well, first of all, we still have so many things to discover. So every time that we do something new, we discover new things. Typically, science makes progress first with technology. A new technology arrives, we apply it, and we find new things. So technology first, and then discoveries, so big discoveries. And then there's the third phase, which is interpretation, knowledge. And that arrives later. So typically, knowledge arrives in unexpected ways. For example, the development of vaccines against COVID occurs thanks to basic research, a basic research that was carried out 20, 30 years ago on bacteria. And this is pure basic reach. Nobody saw any application for it. They were just curious to understand how this could work. And now, you know, how this technology has really helped us thanks to the vaccines. And so this is just one of the examples. But we have also many other examples. Even the questions do not change a great deal, but it's technology that changes. So today, the questions, the issues we try to explore are the same questions that Darwin was asking 200 years ago: diversity, the relationship with the ecosystem, and so on. He was trying to understand how we, just as we do, the same things basically, only that we have more means, we have more technology to discover new things. And so we understand the functioning of the system as we go along. But there's also a part which consists of identifying the various pieces of the ecosystem. Then you need to build the ecosystem with the various pieces and then explain how an ecosystem works. And very often we're still there, we're still collecting the bits and pieces, so we don't really understand the system if we don't have all of the various pieces. It's like, doing a puzzle. If you can't miss some of the pieces. This is the way I see our progress with science. I don't know if there are any questions coming from the audience. I can maybe also put some videos on if you want. Would it be possible to have the video back on? The presentation?
Più simpatico, ok. Vi faccio vedere un video preso in Antartide.
This is a video in Antarctica. So if you don't want to listen to my answers or questions, you can have a look at this. Yes, we have a question there from the second row. Biodiversity, also adaptability to changes. Sorry, we couldn't hear very well. Yes, we couldn't hear very well, unfortunately. You said that in the oceans there's more biodiversity than on land. This biodiversity can better adjust to changes such as, I don't know, the oceans warming up, or is it a delicate system and so maybe the opposite could hold true? Well, it depends. There are some species which are exotic species that would disappear straight away. There are other species which are instead much more resistant. There are species which are very resistant, such as certain animals that we see in cities, such as pigeons, cockroaches, that are easily adjust to us, whilst others can't. So it depends on what species we're talking about. Sorry, it's not audible here. I don't think the mic is on. If I understood correctly, he's saying the relationship between biodiversity of a system and the functioning of a system. Now, for instance, if we take the example of the forest in Amazonia, there is an enormous biodiversity. There is also productivity in terms of photosynthesis, which is very, very high. But alongside this, if we see, for instance, a maize field, single culture, so very, very low biodiversity. And this can have a high productivity, just as much as a tropical forest. And so the relationship between biodiversity and productivity depends, let's say. It's not always the same. It depends on other things. And researchers are very active in trying to understand what the relationship is, because this would explain a lot about the importance of biodiversity, because we need to protect it, because biodiversity is very precious, not only because it's wonderful to go out for a walk in the mountains, but also because there are the services rendered by an ecosystem that make it extremely precious. And this is very important to understand and to understand what will happen in the next few years and protect the different ecosystems in the best possible way. I don't know if this answers your question. Do we have any other questions? Yes, back there. Anybody would like to go on Tara's expedition? Yes, you have a volunteer who would like to leave straight away. So thank you very much for this presentation, first of all. I just wanted to know, in the Mediterranean, one of the problems of climate change are the alien species. In your tours around the world, did you also assess alien species on a global level? And what's the area where the impact is stronger? Alien species, if you've noticed in the Mediterranean the presence of alien species, and where are they more dangerous? That's a good question, but it's difficult to answer in a simple way. Yes, alien species do exist. Not only because the temperature of water changes, certain species migrate, but also because the opening up of the Suez Canal, for instance, has brought in species from the Red Sea into the Mediterranean. So these movements actually take place. and they're quite frequent. With an increase in the temperature of water, species migrate. Species are very, very sensitive to temperature. We suffer here too because of the heat, and species are, set for a certain temperature. So when it changes, they tend to go away, to escape, looking for a temperature that suits them better. And so this means that species in the northern hemisphere are moving towards the Arctic, and those of the southern hemisphere are going towards Antarctica. And we have data showing the plankton and how it's migrating. Fish are migrating, and fishermen have to chase them, let's say, and look for them in different places. And this has also been verified with strong evidence. For the Arctic and Antarctica, this is a problem. For the species which are in these extreme ecosystems, they can escape nowhere because the temperature goes up, and we have seen with our analyses that these organisms that, for instance, in the Arctic, the ecosystem is a world on its own. We don't have the same organisms which adjust better to the cold, but they're really different organisms, such as penguins in the south or polar bears. And this is very sad to imagine that with the melting of the ice, this ecosystem will basically disappear in the next decades, probably. Different simulation models envisage that it will be possible to go sailing at the North Pole in the summer. By 2040, this will be possible. All of the models are saying this. All of the simulation models are saying this. So I would believe this is a reliable piece of information. It's really sad that these organisms, these beings, will probably, or a part of these, will disappear. But life changes. History, the history of life on Earth is a history of changes. There are mass extinctions, 95% of life disappears, but then it comes back, life comes back. There are other organisms, there are other ecosystems that appear, but life wants to resist, wants to continue, and this is what makes a difference with physics, with chemistry, that life is so special, and so there will always be, or for billions of years, life will still be here, life on Earth, on the planet. Probably will be very different from the life that we have today.
Thank you. It's a matter of understanding whether we'll be there. Let's not dwell on these details, Chris says. Thank you very much for your presentation and your work. I wonder, the study of genetic variability in prokaryotes taught us a lot about man's nature and allowed us to find genes that allow us to change a genome, such as the CRISPR system. In such a variability of life in ocean, did something like this emerge? Is there something that can change our life? Very good question. Yes, the answer is yes. Discoveries were made when checking our data, and we did not expect this. You spoke about CRISPR-Cas. Genome editing techniques. we discovered, not us, of course, other researchers, discovered that there are new systems for gene editing in sea bacteria. These bacteria are even more powerful, more targeted, different properties, and they can be used for CRISPR-Cas. This is also true for pharma molecules, industrial molecules, new kinds of glues or adhesives have been found that can be very useful in the water, enzymes that can work at low temperatures and that we can use for washing machines to clean the laundry using cold water instead of hot water. So plenty of discoveries were made, and this is but the beginning of these kinds of discoveries and research. So
deep well.
It's a deep well of information. I forgot to mention this in my presentation, but all what we do is open source. Once the data is published, it is accessible to everybody. And this allows for these kinds of discoveries, and this is extremely important. It is not just something that we keep for ourselves. This is available to everybody. You too can have access to this data. You can look for your favorite microorganism, favorite gene, see where it is located. So this is very important that I forgot mentioning during my presentation. Very important indeed, that we should be reiterating the fact that by destroying biodiversity that we don't even know, we are destroying treasure troves that would be very important for us, and this even before knowing them. There is time for another question. There is no question. A very last question on Tara Ocean's schooner. There is always one artist and one journalist. This is what you said. The artist is an old tradition, because even Darwin had a person drawing. And the journalist, of course, is there to report. What was their relationship with the scientists? Why did you take this decision? How did it go? That when you carry out a research, there is someone else who is reporting it. Well, the experience was very positive, though sometimes it was difficult. It was difficult to explain science to someone who is not a scientist. Sometimes it is difficult, of course. It is difficult to allow sailors and scientists to interact, or scientists and artists to interact. So 14 people on board, 6 sailors, 6 scientists, 1 artist, and 1 journalist. We are in the middle of the sea. We must live together for months. So, on board, there is a very special ecosystem. It is beautiful, I must say, helpful to scientists, because it helps us understanding how to explain complicated concepts. And journalists and artists are supported because they are helped to understand what is science. So it is a very rich kind of interaction. We all appreciated it. Even the relationship with the world of sailors, they are another form of professionals, a highly professional job to tour the world with all kinds of weather, with all kinds of conditions. It is a job, very difficult job. They were very good. They too were very curious, I must say. So it was a crew of people who collaborated well. Very rich experience, I must say. And the role of artists cannot be underestimated. To explain science, to illustrate science is key if you do it artistically. Bergamo understood very well, because you also have artists who make shows during this festival of science. This means that you fully understood how art is important to explain science. And this is very important. So there is also a human biodiversity on this schooner. Very interesting project. You can follow this project online. Thank you, Chris Bowler. Thank you for being with us. Thank you.
Thank you. Thank you so much. Bene. Thank you. Grazie.